In this unit, we're going to look at RNA and its function inside of the cell. RNA is required to make proteins from the DNA double helix. Proteins are required for nearly every activity and chemical reaction within the cell, so this process is vastly important. As you look at this process more in depth, remember these basic steps. DNA stays inside of the nucleus. RNA comes into the nucleus to make a copy of DNA. Once RNA is out of the nucleus, then proteins can be made. RNA, ribonucleic acid, is the other type of nucleic acid found in cells. RNA differs from DNA in 3 ways. Looking at this, on the image on the left, you have DNA. On the right, you have a single strand of RNA. DNA is a double helix, whereas RNA is single-stranded and is a helix. Deoxyribose is the 5-carbon sugar, and deoxy would refer to the lack of oxygen coming off of the carbon. RNA uses ribose sugar, and it has the oxygen coming off of the 5-carbon sugar. The last difference is the use of uracil. Uracil is going to replace thymine. So because of base pairing rules, adenine will base pair with uracil, and cytosine is still going to base pair with guanine. So the 3 differences again: RNA uses ribose sugar instead of deoxyribose, RNA is single-stranded, which DNA is double-stranded, and RNA uses uracil instead of thymine. Now there's 3 types of RNA, each with a different job in the cell. The first is mRNA, known as messenger RNA, and this carries copies of the DNA sequence outside of the nucleus. Next you have ribosomal RNA, which is known as rRNA. This forms ribosomes. The last type of RNA is transfer RNA, or tRNA. This carries amino acids to form protein chains. DNA is essential to a cell's survival because DNA contains the genetic information. To protect DNA, DNA remains trapped inside the cell's nucleus except during cell division. The job of mRNA is to make a copy of the DNA sequence so the message could leave the nucleus without endangering the original genetic blueprint. The process of making mRNA copy from a DNA template is called transcription. During transcription, an enzyme called RNA polymerase unwinds a small section of DNA strand, then uses one side of the double helix as a template to make an mRNA. RNA copies specific genes by finding promoter sequences at the beginning of each gene. What's going to happen is there will be an RNA polymerase, and it looks for the promoter, a start. Once the RNA polymerase finds that promoter sequence, then it's going to go and match base pairs. The template for making mRNA is easy to follow because of base pairing rules, and this was set off by Chargaff in 1950. A base pairs with U, C base pairs with G. Whenever the DNA sequence contains adenine, and uracil is made rather than thymine. So if the DNA sequence is following ATTGACAGTCGA, the copied mRNA strand would be U, A, C, U, G, U, C, A, G, C, U. Now, not all of the mRNA is going to be used. Not all the newly created mRNA is required to code for a protein. Some of the mRNA is spliced out because it's nonsense or unnecessary for that gene. These unnecessary sections of mRNA are called introns and are removed prior to the next step in the process. Introns, portions of mRNA that are removed or spliced out. The exons are the remaining portions of mRNA that will be used to make a protein. So the exons are portions of mRNA that will be used to make the protein.